Welcome to the Honeybee Watch Symposia series. Hello from all over the world. I'm Steve Rogenstein, the project director for Honeybee Watch and your host today. And people probably know Arrigo by now, our science director. He is behind the scenes. He's our master of technical issues and resolving them, and also the moderator of the Q&A that will be at the end of today's program. So today we'll be hearing from 3 PhD students who are at different stages of their studies, and they come from different universities and different countries, but each of them specializes in studying free-living honeybees. And today we're going to be hearing about their areas of focus, what observations they've made so far, any current data and findings that they can share, and more, hopefully anecdotes too. And once we've heard from them individually, I'll moderate a panel discussion between the 3 of them. And then at the end of the program, Arrigo will lead the question and answer session with the audience. So just to start, I'll briefly introduce Honeybee Watch for those of you who are unfamiliar. We are an international coalition tasked with better understanding how free-living honeybees survive via natural selection with little to no human intervention. And by free living, what we mean are bees that have chosen where they want to live, whether that's in a tree cavity, in a log hive, in a hole in the ground, or in human-made structures such as building walls, chimneys, or even abandoned beehives. And our aim is to find nature-based solutions to lower mortality rates of managed colonies, to increase beekeeping's sustainability, and ultimately to protect any threatened wild populations around the world. In our 5+ years, our research partners and citizen scientists have identified more than 2,500 nest sites on 5 continents, and that number keeps rising. One project that we have been working on is coming to its conclusion. After 2 years, a task force that we put together to reassess the red list status of Apis mellifera in Europe, a collaboration with IUCN, the International Union for the Conservation of Nature. We have some game-changing recommendations that will be announced next month, so stay tuned. It's very exciting for us after all this work that we've done on it. We're also part of a consortium that applied for and was awarded a 1.1 million euro grant from Biodiversa Plus and the European Commission. And that's to extensively study free-living colonies in France, Ireland, Poland, Portugal, and Sweden. And that's a project called FreeBee, which you can learn more about at free-bee or free-bee.eu, as Arrigo just dropped into the chat. And our primary mission of forming this global coalition, one of the kind of initiatives that we embarked on 3 years ago was to start the Symposia series. And it's to connect the community, including you, to one another. So now you kind of have the basic skeleton of what Honeybee Watch is all about. And now I'm going to contextualize what the Symposia series is about, especially for our newcomers. First of all, we have something like 390 people who have signed up for today's event. So super exciting. I think that that's the best attendance we've had so far. So thank you for your support. So we launched a series 3 years ago as a means to bring together the free-living honeybee community. And it was an opportunity to link scientists and experts from around the world so that they could facilitate the exchange of expertise, data, best practices, so that you, the audience, could be introduced to their work and you could link to one another. And obviously, we're committed to exploring and discussing challenging topics that we have faced and other researchers in this community have also faced. So in the first year, we explored monitoring protocols, for instance, which resulted in an open source document that's free to download from our resources web page, which Arrigo already put in there. And there, like I said, you can get all of our other videos, but also there are articles on free-living bees, natural beekeeping, tree beekeeping. There are videos, there are journal articles, there are posters. So explore that resources page if you want to get more information about these topics. In the first year, we also discussed data sharing and data rights and how to define wild in the context of free living colonies, which is a difficult challenge because wild can be construed in so many different ways when related to bees. In our second year, we explore bee lining, tree cavities, whether wild honeybees are wild bees too, and best practices for communicating and recruiting for citizen science projects. And then this year, we kicked off in March with an introduction to Free Bee. That's that study that I referred to before from Biodiversity Plus. In June, we talked about free living honeybees in Africa. And then in December, to round out the year, we're going to be exploring bee-related policy issues. And if you want more information about the series or to register for December or for the past so that you get the links to the videos, you can sign up on our events page, which Rigo just dropped into the chat. So that brings us to today's program, which we're calling the latest buzz, current research from Honeybee Watch Partners. With that, I would like to introduce our next speaker, Alex Valentine. She is wrapping up her studies in Ireland, but calling in from the UK today. She studies under Grace McCormack at the University of Galway, where she's been working for several years on the outside the box research study, which questions how Irish's free living colonies are able to survive unaided outside of conventional beekeeping settings. And it's the first countrywide study on free living colonies in Ireland. So this is super exciting stuff. And she and Grace and Rigo and Keith and Chiara have all been working on this for, I believe it's 8 years already. So there's lots of information that's coming out of Galway. So you should definitely be looking out for publications that reference these. So because Grace is so integrally involved in Honeybee Watch, obviously we were quickly introduced to Alex and have been working together either directly or indirectly on a few projects ever since. So just a shout out to Alex. She was instrumental in organizing the freebie proposal for Biodiversity Plus. So thank you. And she also spoke briefly about the research at Galway during the Speed Bee Talks presentation at the 2023 Learning from the Bees conference. So we've heard a little bit about what she's working on, but thankfully today we're going to hear more.
Thanks very much for the introduction. As Steve was saying, my name's Alex Valentine. I'm a PhD student finishing my thesis at the moment at the Galway Honeybee Research Centre with Arrigo and Grace and Keith and the team. And the title of my thesis and the title of my talk today is Diversity and Adaptation in Irish Honeybees. And that's kind of been my sole focus for the last 4 years. So I'll just touch briefly on why wild honeybees are so important. So Apis mellifera, or the western honeybee, has obviously evolved into many different subspecies or races across its distributional range. The subspecies that we're most interested here in Ireland is Apis mellifera, but genetic variation in general is necessary for evolutionary success, and all of these Apis mellifera subspecies are adapted to their local environments in different ways, and this local adaptation leads to much greater local fitness. But there's also then a side component here that selection is also strongest on wild organisms, so therefore wild honeybees in general, meaning that we can answer lots of questions that are plaguing the managed population with answers that the free living population have already worked out for us. So as Steve was saying, the honeybee research at the University of Galway commenced back in 2014, with Grace has kind of been integral and vital to that process the entire time since 2014. Arrigo and I have only been part of the more recent project, but we are very grateful for all the work that kind of came before us. So back in 2014, the central research questions for the team were to understand if we had any pure Apis mellifera left in Ireland, to possibly establish a research or a teaching apiary and develop a breeding program for varroa tolerance or resistance, and answering the question of whether we had any wild AMM present in Ireland at all. And they came up against a lot of assumptions that feral colonies or free living colonies had been completely eradicated in the UK and in Ireland for a variety of reasons, but most of the time it was attributed to the Isle of Wight disease or World War One, the impact of World War One. And lots of people were kind of saying to the team, nope, there's no wild or native colonies left. They've all died for varroa or hybridization or something else. But black bees are native to the island of Ireland, and a lot of these assumptions were incorrect or were found to be incorrect when the team published the results of their first wave of studies, looking into the genetic integrity of Irish samples and looking at Irish free living colonies. The graph here at the bottom is just a graphic from one of those papers, and I'd just like to showcase within the green circle we have all of the Irish samples that were studied for that particular paper, and the blue and orange is just the comparison of M lineage or C lineage honeybees, and in Ireland we're interested in only M lineage. So we can see even from our initial investigations, the majority of what we have in Ireland is thought to be pure M lineage. So now that we'd confirmed that Apis mellifera mellifera was the native subspecies in Ireland, and we had a really widespread and pure free-living population identified at the same time, we were also then able to track the survival over those first 4 years and found some really good persistence over the threshold expected for varroa. And this kind of led us to formulate the hypothesis that we have one of the largest stable remaining populations of AM mellifera in Western Europe. Then, combined with some ongoing research that Grace was involved with, we were able to document that levels of introgression were still increasing into Ireland. So imports of C lineage subspecies were threatening the genetic integrity that we have in Ireland. Led Grace to come up with the idea of developing a new wave of project and a new bout of research questions. And this was where me and Arrigo were given the opportunity to be involved with the team. And when we started looking for evidence of the risk of imported bees to the unique Irish honeybee. We also wanted to look at factors that might be influencing the wild honeybee survival that we had been seeing. We were then looking to investigate relative fitness of wild versus managed bees as well, to kind of challenge the assumption that free-living bees aren't good for managed bees, or that they shouldn't be utilised because they do represent some quite important resources. And also to develop some sustainable local beekeeping using the research and teaching apiary that they'd set up in the previous wave of projects. So now that we had a large free-living cohort identified across Ireland, we had 76 nests identified in 2020. We then increased our monitoring efforts, and as a result of the partnership with the National Biodiversity Data Centre here in Ireland, we got mass citizen science participation, and now we have over 750 nests identified in 2025. Of those 750 nests, we're monitoring approximately quarterly with 175 of them. And most of those that we're focusing in on are being found in building or tree cavities as quite common nest sites in Ireland. We're also seeing quite good survival of these colonies as a result of the regular monitoring. We're able to track the survival of the colonies really in detail. On the left here, we have all of our 2024 and 2023 check-ins with each of our colonies. So each one of the small squares is a report. Green meaning that the colony is active, red meaning inactivity or the colony has died, nest sites become empty, lots of different things. But we're seeing overall, when we look at the overall survivorship in the free living cohort, we're looking at about 80% survival over winter, which is excellent. And the more in-depth stuff that we're starting to break into now, we're using the genetic data from these colonies as we're regularly monitoring them, we're regularly sampling them as well. And we've been kind of developing ways of testing, comparing samples year on year to see if they're still related, or to see what's happened. Has the existing genome that was already, or the existing genotype, has it been replaced? or is this a second or third generation that we're dealing with? So from these comparisons, we can quite easily tell which colonies are still related, still the original colony persisting over multiple years. This kind of analysis gives us these answers in really clear detail. So in green there, we have all of the sites where we believe it's the same lineage persisting over 3 or 4 years. Then we have some cases where it's a second degree, so it could be that the existing queen has been replaced slightly, or it could be a second generation daughter or something like that. We can also see then cases where they're completely unrelated, so we're quite confident that the colony has died, and then since then a completely different lineage has moved into that nest site. In some cases where we've been able to track, where we've been getting samples every year continuously, we can also see the progression of this relatedness dissipating. So we started down here in 2024, and we can see when we compare that to the 2022 or the 2021 samples that the relatedness does start to go down over the generations. In addition to this, we performed some genotyping analysis across 500 samples that we got from local beekeepers. We run these through a 62 SNP panel to identify how many were AMM, how many were introgressed, what was the exact risk to the genetic integrity of the Irish honeybee. We found that 381 of these were Apis mellifera mellifera, or pure samples, and 124 were introgressed or hybrid, giving us a 25% introgression rate, which I've included the link to this paper at the end, and if you wish to, there's some more information in there about how we've been seeing these introgression rates increase over time. And that was another huge motivator for us to conduct this research in the first place, is we were seeing visually that the introgression was increasing and that we were seeing that the risk was getting worse, and that encouraged us to start this research in the first place. And as a result, we also were able to get average M lineage values per county, which can help us paint a picture of current hybrid hotspots and identify routes of import of introgressed colonies or queens, and identify areas of highest risk for the genetic integrity. So my most recent project has been looking at pathogen analysis in these Irish free-living samples as well. Here we have the prevalence of 18 key microparasites or pathogens involved in poor colony health or decline. We tested these using qPCR and some in collaboration with some researchers in Spain. And from this, we were able to see the Irish samples have very high prevalence of Lopmaria passim, which is a gastrointestinal parasite, thought to be quite lethal, but it's very similarly related to Crithidia mellificae. And when you look at Crithidia or Lopmaria tends to be the less severe of the two. So it's definitely a positive that we didn't see any Crithidia mellificae here, but still slightly concerning that we have such high levels of, or high prevalence of, Lopmaria passim. As to be expected, we saw quite high levels of deformed wing. This is considered to be the most prevalent honeybee virus, so aligned perfectly with our assumptions. We were expecting to see lots of this in both the free-living and the managed population. We saw variable levels of European foul brood, or Melissococcus plutonius, detected. It was around 10% in the managed samples that we assessed, and then around 25 in free-living samples, but there was no significant difference between these two prevalences. It's just was quite interesting to us that they were so variable between the two. We also saw low levels of chalk brood, which quite surprised us given the climate in Ireland is quite damp and wet, we were expecting to see quite high levels of chalk brood, which we didn't. We also didn't see any American foul brood, any Crithidia mellificae, and no Nosema apis. We saw quite high levels of, or quite high prevalence of Nosema ceranae, and this was the only pathogen that we found that differed significantly in the prevalence between free-living and managed bees, being significantly higher in managed untreated colonies than any of the other cohorts that we assessed. If we visualize this on a network and we look at the communities of infectious pathogen or infectious agents, we also saw no significant difference in the number of pathogens or the patterns of infection between free-living and managed colonies. But we did see one of our observations is that managed colonies show a higher diversity of pathogen communities. So we can see from this graph here, we have, I think, 9 different patterns of infection for the managed treated colonies and only 6 for the free-living colonies. So there is a slight difference in the infectious communities, but not enough to generate a significant difference in the samples we've assessed so far. When we then look at the nest site diversity of some of the colonies that we're monitoring now, our previous observations found that we were looking at a mix of building cavities, tree cavities. That's still very much the same, or very much the same even now. The majority of the colonies that I assessed for pathogen prevalence were located on pasturized land or discontinuous urban land, and evenly split between the two, really. Roof cavities and tree or log cavities are still prevalent nesting habitats, which gives us good evidence for persistence of colonies in urban environments as well. It's not just tree cavities that are necessary for them to be able to survive. We're seeing quite good survival and good health in general in colonies that are persisting in urban spaces as well. So to kind of wrap all of that together, we think over the last 8 years, we've found evidence of sustained survival in Irish free-living colonies, and now in particular in the face of pathogens thought to be responsible for poor colony health or decline. My pathogen analysis has led us to the assumption that free-living colonies in Ireland anyway are indistinguishable from managed colonies in terms of their patterns of infection and occurrence of pathogens, challenging the observation that they're detrimental to the health of managed hives. So we don't really believe that they are detrimental to the health from our analysis. The health or the pathogen occurrence anyway is fairly ubiquitous between the two. And this lack of difference could indicate that we have one continuously flowing pangmictic, continuously mating population, with a significant urban population maybe facilitating that and facilitating continuous mixing and local adaptation that we would expect to see when they're surviving. Thanks to everybody for your attention. I've left my email at the bottom here if anyone wishes to get in touch. Again, thanks to Arrigo and Grace and Keith and the team for teaching me all of this. And I can't really believe that 4 years is over already, but I have had the best time and I've enjoyed doing this research that entire time.
Well, thank you, Alex. What an amazing opportunity for you to be on the team that's really leading the field in research on free-living colonies. So congratulations to you and Grace and Arrigo and the rest of the team. What's the takeaway for me from what you just presented is it doesn't look like free-living colonies are suffering from high levels of pests, pathogens, diseases, etc. And at Honeybee Watch, what we're trying to understand are these attitudes and misperceptions about free-living colonies being mite bombs, for instance.
Yeah.
And if we can convey to the community and beyond that that is not the case, I think that we will have succeeded. And you guys, like I say, are leading the path. So bravo, and thank you for sharing all this.